Okay, so let's start, Marcelo. Good afternoon, everybody. So it's a great pleasure to welcome Professor Marcelo Hill. Professor Marcelo is the head of the Department of Immunobiology at the Faculty of Medicine and leads the Laboratory of Immunoregulation and Inflammation at the Institut Pasteur de Montevideo. His research focuses on the mechanisms that regulate immune responses in cancer, inflammation, and immune-mediated diseases, with a particular interest in innate immunity, inflammation biology, and cancer immunotherapy. Over the years, his group has made important contributions to our understanding of how immunoregulatory pathways shape anti-tumor immunity and influence responses through immune checkpoint blockade. More recently, his work has highlighted the therapeutic potential of targeting TMEM176B as a novel strategy to enhance anti-cancer immune responses. Today, Professor Hill will present his seminar entitled Pharmacological Inhibition of TMEM176B, Reprograms Th17 cells to enhance PD-1 blockers in cancers. Thanks for being here, and the word's yours, Marcelo.
Thank you, Vinicius. I'm very happy to give this talk. And I remember that some years ago I was invited by Alessandra, and I think that it was at the very same place that I gave a seminar, and it was I have very nice remembers from that time too. As Vinicius said, I'm going to talk about this immunoregulatory protein, TMEM176B, which is the focus of our work in the last years. And now we are trying to characterize this protein as an important regulator of Th17 biology, and particularly in cancer and under immune checkpoint blockers. So these are the disclosures. And well, as you know, anti-PD-1 has increased the overall survival and progression-free survival in several cancers. But we know to improve our understanding on the mechanisms in order to overcome primary and second resistance, as well as to better control immune-related adverse effects. And we know that PD-1 blockade primarily targets CD8 T cell exhaustion. And in the next slides, I'm going to focus a little bit on this complex process. And we also know that gut microbiota determines the responses to PD-1 blockers. And one of the intriguing things is that Th17 cells are known to modulate exhausted CD8 T cells, but their role in tumor immunity is quite controversial, and we will analyze this because of this effector and regulatory polarity within the Th17 cells. So one of the questions that has driven our work is whether intestinal Th17 cells can control T cell exhaustion within the tumor microenvironment, and research that I'm going to show you dealing with this question, and by studying TMEM, try to answer to this question. So, as you know, too, one of the main mechanisms of PD-1 blockers is to either to prevent CD8 T cell exhaustion or to induce
Marcelo, sorry, we have an issue with connection.
Of exhausted CD8 T cells, and by doing so, is tumor microenvironment.
Marcelo, can you hear me? O pessoal no Rio de Janeiro está conseguindo ouvir ou está travando aí com vocês também?
Gente, ouve vocês bem.
Marcelo, I think we have an issue with the connection. Can you hear us? Acho que caiu. Eu acho que é lá.
Acho que voltou.
Eu acho que caiu.
Can you hear me now?
Yes, yes, now it's better.
Okay, good. So sorry. So I was telling you that these exhausted T cells are a distinct cell lineage, which has been described in chronic infections as well as cancer in mice and humans. So the main hallmark of CD8 T cell exhaustion is a progressive loss of effector function and proliferative potential, and classically inflammatory cytokines such as interferon gamma and TNF-alpha are downregulated in these exhausted T cells. So these cells are associated with increased expression of inhibitory receptors such as CTLA4, PD-1, LAG3, TIGIT, and many others. And particular programs at the metabolic, transcriptional, and epigenetic levels have been associated with this exhaustion state. And we know that there are several subsets within exhausted T cells. So one of them is the stem-like or also called progenitor exhausted T cells. And these cells are very important because they are expanded when you block PD-1. So when patients are treated with PD-1 blockers, this particular subset is induced to proliferate. And then this can differentiate into transitory exhausted T cells, which express granzyme B and have some capacity to kill malignant cells. So they are not as good as effector cells. That's why they are exhausted, but they retain some capacity to kill cells. And this capacity to kill malignant cells is completely lost in the terminally exhausted cells, and we can identify this subset with some of these markers that are shown there. So the question is, we need to improve anti-PD-1 therapy, and one of the pathways would be to trigger CD4 help, right? Because we know since some years now, thanks to this paper from Rafi Ahmed group, that CD4 cooperation can rescue exhausted CD8 T cells, and this has been shown in viral infection models. But one of the main questions is, which is the right subset? We may intuitively say that Th1 cells would be important partners to rescue these exhausted T cells, but we want to see in the next slides that this is not necessarily the best option. And we also need to define pharmacological targets. I mean, how to specifically trigger CD4 cooperation to rescue CD8 T cell exhaustion, that we have a problem there by how to target pharmacological interventions. So we know from different papers and different models that Th17 cells can outperform Th1 cells in controlling, in inducing tumor immunity. So these are adoptive cell transfer experiments in a melanoma model in this recent paper. And you can see that Th17 cells can control tumor growth with much better efficacy as compared to Th1 or even Th9 cells. And in the low part of the slide, you can see another work where you can see that a specific subset within Th17 cells called Th1-like Th17 cells can control tumor growth in a better manner compared to Th17 or even Th1 cells. So if we will be able to trigger Th17 cooperation, that will be a potential way to control tumor progression and to control CD8 T cell exhaustion. So just to remember that Th17 cells are defined as IL-17 producing CD4 plus T cells. They also produce other cytokines such as IL-22, IL-21, 2, and ROR gamma T is the main transcription factor which drives their differentiation. And classically, Th17 cells are known to control extracellular bacteria, to promote mucosal homeostasis in the lungs, but also in the gastrointestinal tract and genitourinary. And they can also mediate autoimmune damage. We can differentiate these cells in vitro with two different cytokine cocktails. If we use IL-6 plus TGF-beta, we are going to produce homeostatic or regulatory Th17 cells. Whereas if we differentiate CD4 naive cells in the presence of IL-6 plus IL-1 beta plus IL-23, we're going to generate inflammatory or pathogenic Th17 cells. But what do we know about Th17 cells in cancer? So here the scenario is quite complicated because there is literature like this paper some years ago showing that IL-17 and Th17 cells can be associated with a clinical benefit when melanoma patients are treated with anti-CTLA-4 and anti-PD-1 antibodies. But then there is also this kind of paper from Padmani Sharma's group, where they have shown that in prostate cancer, in the osseous metastasis, you can have very low response, and this is associated with infiltration with Th17 cells. However, if they add to PD-1 blockade anti-TGF-beta antibodies, then the infiltration by Th17 cells is going to be clearly decreased, and infiltration by other T cell subsets will increase, and there you have a good response. So Th17s may play, on both teams, where anti-tumoral team or pro-tumoral team. So to sum up a little bit this complex scenario, we can say that Th17 cells are very plastic and heterogeneous. So on one side, you can have non-pathogenic or homeostatic Th17 cells, which produce immune regulatory cytokines such as IL-10. But on the other side, you can have Th17, which are inflammatory, pathogenic, or Th1-like, where they start to produce inflammatory cytokines such as interferon gamma or even GM-CSF. And these cells, from a transcriptional metabolic point of view, mimic Th1 inflammatory cells. And we know that there are particular transcriptomic, epigenetic, and metabolic programs that are interrelated and are controlling the differentiation within this continuum from homeostatic regulatory Th17 cells to inflammatory or pathogenic. So there are certainly, physiological checkpoints that will control this transition. And if we can describe them, we could have a pharmacological way to control differentiation from the regulatory to the pathogenic or inflammatory side. And we know that these differentiation pathways are really complex. They mainly happen in the mucosa, particularly in the intestines. And since some years now, it has been described stem-like Th17 cells, which is TCF1 positive. And these stem-like cells can differentiate either into anti-inflammatory Th17 cells, which produce IL-10, or alternatively, they can differentiate in the presence of IL-23 into pathogenic inflammatory Th17 cells, which express Tibet interferon gamma. And these inflammatory cells have been described to migrate to the central nervous system and mediate damage in EAE in the EAE model. So these stem-like cells have, the choice either to differentiate into bona fide regulatory Th17 cells or pathogenic Th17 cells. But what happens in cancer? So these differentiation states really change across the malignant transformation. So during carcinogenesis and transformation, these intestinal Th17 cells can differentiate into inflammatory cells, and these Th17 cells are known to be very important, to induce transformation, and this has been described very clearly in colorectal carcinogenesis. But then once the tumor is established and you reach advanced stage, then these Th17 cells can again differentiate into regulatory or homeostatic cells, and in that case, impair antitumoral immune responses and even promote epithelial-mesenchymal transition, which is associated with metastasis in colorectal cancer. So what can we do from a therapeutic point of view? The idea is that at these stages, you may, if we can identify checkpoints within these cells, we may trigger differentiation into regulatory cells to avoid this inflammation-inducing carcinogenesis. And on the other side, if we can pharmacologically manipulate these regulatory Th17 cells, we may transform them into effector or inflammatory antitumoral Th17 cells. But so far, these are just hypotheses. And I want to introduce another player here, which is a different one from the homeostatic Th17 cells, which are the so-called ROR-gamma-T positive Tregs, right? So as I was telling you, these stem-like Th17 cells can differentiate into the inflammatory Th17 cells or the homeostatic ones. But these homeostatic or regulatory Th17 cells producing IL-10 and expressing the transcription factor CMAF can even differentiate into ROR-gamma-T positive Treg cells, which express at the same time FOXP3 and ROR-gamma-T. And it is poorly understood whether these cells differentiate only from these homeostatic Th17 cells or whether bona fide Tregs, FOXP3 positive Tregs, may also differentiate into these ROR-gamma-T positive Tregs. So as you see, the scenario is very complex because of these players and the plasticity and heterogeneity of this system moving from homeostatic to inflammatory players. Some years ago, Laurent Vogel team in France described that during dysbiosis, these ROR-gamma-T positive Tregs may play an important role. So during dysbiosis, these cells are no longer retained in the guts, but they migrate to the tumor. And there, within the tumor microenvironment, these ROR-C positive Tregs can impair an immunity triggered by anti-PD-1, and they have also shown that these mechanisms are relevant in humans in lung cancer patients treated with anti-PD-1. So far, the idea is that if we could be able to identify druggable molecular switches controlling antitumoral polarity of Th17 cells, we may have some opportunity to trigger tumor immunity and improve the outcome of PD-1 blockers. So here's where we speculated that this protein, intracellular protein TMEM176B, could be a potential checkpoint controlling Th17 polarity. And this is a paper from Dan Littman group in 2012, where they described that only about 11 genes were directly controlled by ROR-gamma-T cells. And among them, there were some known genes to play a role within Th17 cells, but there were some others with unknown function. And among them, we found TMEM176B and another member of the same family, TMEM176A. So we know that TMEM176B, which we'll also call TORID, which stands for tolerance-related and induced, is a CD20-like molecule which is highly expressed in myeloid and ROR-gamma-T positive cells. And some years ago, we have shown that this protein is an intracellular immunoregulatory non-specific cation channel, which is localized in the Golgi, endosomes, and phagosomes. And we have also shown that TORID plays a role in controlling inflammation activation and T-cell exhaustion in coronavirus disease. And we have also shown that TMEM promotes tumor progression and immune checkpoint blocker resistance by blocking inflammation activation. And Alessandro's team has shown some years ago that single nucleotide polymorphism coding for a substitution within this channel is associated with good prognostic in colorectal cancer patients. So this is some data on human colorectal cancer. So we have shown this association within colorectal tumors where high TMEM expression is associated with poor overall survival. And these observations were confirmed by Alessandra's team, first showing that high expression was also associated with this poor overall survival. But then Alessandra's team added some very important information. They showed that this single nucleotide polymorphism was associated here in red with good prognostic and good overall survival, and they associated this expression with with high inflammasome activation in peripheral cells and with low expression of this channel. And we also know that there is a causality between the expression of TMEM and colorectal cancer progression, because if we use different colorectal cancer cell lines in wild type or TORID knockout or TMEM knockout mice, we can see that deletion of this gene is associated with control of tumor progression for both lines. And if we add a TMEM inhibitor such as BK, we can even improve the antitumoral efficacy of anti-PD-1 antibodies. And in humans, we know that TMEM is associated with resistance to immune checkpoint blockers. This has been shown in breast cancer. There is a recent paper in Nature Medicine showing that in hepatocellular carcinoma, TMEM176B and TMEM176A are associated with early progression. Sorry. Okay. And then we have also shown in the right part of the slide here that in melanoma also TMEM is associated with increased expression on treatment within resistant patients to PD-1 blockers. So this clinical data encourage us to find, a small molecule to block TMEM activity, and we identify this compound BK8644 as a strong inhibitor of TMEM. And you can see that if we treat mice in the red line with this TMEM inhibitor, we can control tumor progression. Whereas if we do this treatment in inflammasome knockout mice, we completely lost efficacy. And this seems to be a non-target effect, because if we treat the TMEM knockout mice, we have no increased benefit, suggesting that BK is actually targeting TMEM. And this depends on CD8s, and we can improve anti-PD-1s in melanoma models too. So I'm sorry, I want to see if I can show my pointer. Okay, so then our main question was, is there a role for TMEM positive Th17 cells in tumor immunity? Because of this relationship that I commented on the dependence of TMEM on ROR gamma T cells. But so far there is no clear role for TMEM within Th17 cells in a general manner, nor in tumor immunity. So we went to stain in human tumors CD3 cells, CD4 cells, and TMEM, and we found actually some interesting observations. You can see that actually there are CD3, CD4 positive cells that do express TMEM within human tumors, but we can also find CD3 negative cells, probably myeloid cells, which express TMEM, and some T cells that do not express TMEM. But so the main question here is, what is the role of these TORID positive, TMEM positive T cells within human tumors? So then we move to bioinformatic approaches, and we analyze data from this colorectal cancer data set. Within T cells, we found three main clusters, cluster 5, 9, and 13, which had a clear Th17 signature. And these clusters, cluster 9 and 5, express high levels of TMEM176B and TMEM176A. Sorry. And a general analysis of this cluster showed us that cluster 5 and 9 are regulatory ones because they express IL-10, IL-2, and this cluster 9 is a Treg RORC positive cluster, whereas this cluster 5 seems to be, a Th17 regulatory cluster. And when you analyze the cluster 13, you have increased expression of pro-inflammatory markers such as interferon gamma, Th, NKG7, and granzyme B. So this cluster 13 seems to be an inflammatory pathogenic Th17 cell with low expression of TMEM, whereas the regulatory cluster express high level of TMEM. And then we found some interesting clinical pathological correlations. In colorectal cancer, the mismatch repair status is very important from a pathological point of view. And you have the proficient patients and the deficient ones. And the proficient patients are resistant to PD-1 blockers within colorectal cancer patients. And then you can see that both proteins are highly associated with the proficiency status in contrast to the deficient MMR, when these patients are good responders to PD-1 blockers. And when we analyze lymph node metastases, again, within Th17 cells, expression of these markers were associated with metastases in these colorectal cancer patients, suggesting that this TMEM expression within Th17 cells will be relevant from a clinical pathological point of view. So then we wanted to understand a little bit more what is TMEM doing within Th17 cells. So we moved here to mice cells and we performed a single cell RNA-seq study comparing wild type and TMEM knockout cells, and we performed this transcriptomic study. So the first finding was very surprising for us. We found that, we are differentiating here cells, naive CD40 cells with TGF-beta and IL-6. So we anticipated that we would have somehow a homogeneous cell population, but this was not the case. We found actually 4 different clusters, and we annotated them, one of them as a stem-like, which is TCF7 positive, a regulatory one expressing high levels of CTLA-4, and then we have effector subset, although we are differentiating these cells only with regulatory cytokines. And we also have a cluster which have several markers of these Treg RORC positive cells. And you can see that the stem-like have all these markers of stemness. Then the effector cells have expressed interferon gamma, Th, NKG7, and then the regulatory ones express IL-2, CTLA-4, and the other cluster, the Treg RORC positive, express at the same time regulatory and effector genes, as it has been described in the literature. And we also perform a differentiation trajectory analysis by pseudotime approaches, and we found mainly 2 lineages, one which goes from stem cells to effector cluster, and the other one which goes from stem-like cells to the regulatory ones. And if you compare the wild type and the knockout cells, we had a decrease in the knockouts in the regulatory cluster and an increase in the effector one. And this trajectory analysis also showed that in the knockouts we have increased differentiation toward the effector-like phenotype. So all these results are suggesting that TMEM could be driving differentiation from stem-likes to regulatory cells and avoiding differentiation into effector cells. And then to compare the wild type and the knockout cells, we analyze different signatures. And as you can see that in a general manner, here in the knockout, we have increased effector-like pro-inflammatory and Th1 signatures. The opposite was seen in the regulatory-like signatures. The TMEM knockout had decreased regulatory signatures, and there seem to be also important changes from a metabolic point of view with the knockout Th17 cells showing higher levels of oxidative phosphorylation, glycolysis, and TCA cycle. And these are some of the genes that were differentially expressed within the 4 subsets that we found in this study. But then we've showed that functionally and phenotypically these cells are different when we compare wild type and the taurine knockouts. And particularly, if you can see here, the interferon gamma secretion was increased in the TMEM knockout mice. This is a quantification from different experiments. And even if we treat these cells, wild type Th17 cells, with the TMEM inhibitor BK, we can trigger interferon gamma secretion by these cells. And then from a functional point of view, if we use wild-type Th17 cells, we can decrease proliferation in vitro of CD8 T cells, but we fail to control this proliferation when we use the TMEM knockout Th17 cells, showing that the immune regulatory capacities of these cells were lost when we deleted the TMEM gene. And then the transcriptional study suggested that there will be some important metabolic differences within these cells. So we analyzed glycolysis through seahorse experiments and oxidative phosphorylation. And in both cases, we found that TMEM knockout mice showed increased levels of glycolysis and oxidative phosphorylation. And this enhanced glycolysis has been described within inflammatory Th17 cells. So this is in agreement with the phenotype that we are describing. But furthermore, we also showed that if we block glycolysis within these cells, we can block interferon gamma expression. So here we are linking this enhanced glycolytic capacity of the TMEM knockout mice with the increased interferon gamma secretion by these cells in compared to the wild type Th17 cells. But then we wanted to know whether there could be a role for TMEM in tumor models. So we collaborated with Florencia McAllister from MD Anderson, and they have described these mice with these conditional knockouts where they delete IL-17 receptor from epithelial cells. And by doing so, they have shown that there is an increased Th17 cells within the guts, and that these gut-derived cells migrate to the tumor. So you can see here in the graphical abstract, this knockout triggers the dysbiosis, which at the same time trigger Th17 cells that then migrate to the tumor and promote tumor progression. So in this model, we analyze TMEM expression within the cells in the gut and in the tumor. And you can see that both in the knockouts and the wild type mice, within the low TMEM expressors, there was an increase in Tibet and in interferon gamma, suggesting again that within these cells, in tumor models, TORID will be associated with inhibition of this Th1-like phenotype. So we then wanted to know whether, in a more direct way, whether TMEM will control the antitumor function of Th17 lymphocytes. And you can see that if we, in anti-PD-1 treated mice, we adoptively transfer wild-type Th17 cells, and we fail to control tumor growth. But if we transfer knockout inflammatory Th17 cells, then this treatment was had good efficacy and control the development of tumors in these melanoma mice that we're working here. And then to deep these functional observations, we perform the anti-PD-1 plus BK therapy in mice, and you can see here in the red line that anti-PD-1 plus BK controls tumor progression in comparison with the black line where we have only anti-PD-1 alone. But this efficacy was strictly dependent on IL-17 expression, because in the green line here, in the IL-17 knockout mice, this double therapy completely failed to control tumor progression, suggesting that this double treatment, anti-PD-1 plus BK, is depending on Th17 cells to control tumor progression. And then we analyzed within the tumor microenvironment this phenotype, inflammatory Th17 cells, which are interferon gamma positive, CD73 negative, or homeostatic or regulatory Th17 cells, which are interferon gamma negative and CD73 positive. And importantly, we found that if we add to anti-PD-1 treated mice the TMEM inhibitor, on one side, we increase the tumor infiltration by inflammatory Th17 cells, whereas the infiltration by homeostatic Th17 cells was significantly decreased by adding the TMEM inhibitor. And then we wanted to know whether these tumor infiltrating Th17 cells will be arising from the intestinal compartment. So we went to analyze Th17 cells within the colon of these mice, and we found that ROR gamma T positive CD4 cells were increased when we added the TMEM inhibitor, and there was a negative correlation with tumor weight with the absolute number of these cells in mice treated with anti-PD-1 plus BK, but not in mice treated only with anti-PD-1 here in black. And when we analyze in the tumor microenvironment, intestinal derived, which are alpha-4 beta-7 positive, interferon gamma positive, ROR gamma TT cells, these cells were increased in the double therapy, and again there was a negative correlation in co-therapy, but not in anti-PD-1 alone, when we were correlating tumor weight with the absolute numbers of these cells. But to more generally study what was going on the intestinal compartment, we also analyzed through metagenomic studies the intestinal microbiota. So in each column here you have different bacteria species within the gut. And you can see that there was a huge dysbiosis when we compare anti-PD-1 plus BK, when this is compared to anti-PD-1 alone. And even though if we treat these mice with oral antibiotics such as ampicillin, we can impair the antitumor efficacy of anti-PD-1 plus BK. If we do this therapy plus ampicillin, tumors are progressing more. And then the absolute number of these intestine-derived Th17 cells producing interferon gamma is decreased when we treat these mice with ampicillin, showing that intestinal bacteria are somehow involved in this antitumoral mechanism. So just I want to sum up a little bit the mechanisms, but you know, we speculated that these Th17 cells manipulated with BK will be impacting on exhausted T cells. So we analyzed in the tumor microenvironment this progenitor exhausted, the transitory, and the terminal, and we observed that by aiding BK, the TMEM inhibitor, we increase the TPEX, which are very important for the responsiveness to anti-PD-1 antibody, and also the transitory subset, although the terminal subset was not modified. And we perform this treatment in IL-17 knockout mice, and you can see that within transitory and terminal, we can increase granzyme B expression with the double therapy, but this effect was lost in the IL-17 knockout mice, suggesting that IL-17 and probably Th17 were involved in these effects. And if we treat the mice with ampicillin, trying to demonstrate a role for this intestinal microbiota, again, these transitory cells were decreased in the double therapy anti-PD-1 plus BK. And in vitro experiment, we show that the knockout Th17 cells can control terminal exhaustion. So here we are measuring terminally exhausted cells. Wild type Th17 cells cannot modify them, but knockout Th17 cells were able to control terminal exhaustion. So coming back to human data, in lung and colorectal cancer data sets, we observed that these progenitor exhausted, which are very important for responsiveness to anti-PD-1, are negatively correlated to these RORC positive, TMEM positive Tregs, and these cells were increased in resistant patients to PD-1 blockers in a human cohort of lung cancer patients. So just to finish, I wanted to comment you that we can manipulate the human Th17 cells with the inhibitor BK. So if we treat in vitro Th17 cells, we can decrease the differentiation into Th17 cells, and we can also trigger with this TMEM inhibitor interferon gamma expression, probably inducing the differentiation of these cells into antitumoral cells by expressing these antitumoral cytokines such as interferon gamma. And we also performed a molecular study, a docking study, which suggested a potential pose for BK, and we identified the amino acids that were interacting with the compound, and we have functional data with the mutants with these amino acids showing that this molecular mechanism seems to be important, not only for BK inhibition, but also for the loss of function, which is associated with the A134 substitution. Here you can see that this mutant is a loss of function, and we can rescue this function if we play with these other amino acids here. And finally, we think that these polymorphisms can predict responsive of individuals, of human patients to BK therapy. So you can see here the difference in interferon gamma secretion when we treat them with BK versus the vehicle, and within the reference genotypes, you have this response, and then in the loss of function genotypes, you cannot improve this with the compound. So to sum up, we think that by treating the mice with the inhibitor, we are transforming these regulatory homeostatic Th17 cells into effector cells that then will migrate from the guts to the tumor microenvironment, and there they can collaborate with exhausted T cells and improve the antitumor efficacy of anti-PD-1 antibody. So just to finish, let me thank the people in the lab. So Sofia Russo, who is who has been leading this work in Th17 cells and tumor immunity, Mercedes Segovia, which also directs this work with me, and then collaborators in the region and broadly. And to finish, I want to just invite you to this meeting that we are organizing in October in Uruguay, where we have a basic immunology course given by Abu Labas, and then we have some workshop on tumor immunity too, and other settings in immunology. So thank you.
Hi, Marcelo. Thanks a lot for a great talk. So now we are open for questions. No questions from students. Okay, so In Rio? Someone has question, Rio de Janeiro?
No questions so far.
Okay. So I think I have two questions. First, I remember I had the pleasure to be in your lab this year, so we had this collaboration, and I remember that you also identified some compounds that are thyroid agonists. So can you tell us something about the effect of these agonists, and also regarding when should be more indicated to use a thyroid antagonist or agonist, and the toxicity assays and all these things?
Yes, that's a very good point, Vinicius. So what we think is, that TMEM is a kind of a checkpoint within the immune system. We have previously described its role in myeloid cells and controlling inflammasome activation, and now we are focusing on these Th17 and Treg cells. But the main idea is that thyroid is promoting regulatory responses. So in the context of cancer, the main interest is to block it and to trigger anti-tumor responses. But we can also think that in inflammatory diseases, you can trigger TMEM activity and control, for instance, autoimmunity. So we, as you said, we have identified TMEM agonists, and we are now working on these autoimmunity models, trying to see whether we can improve autoimmunity outcome by triggering TMEM activity. And if we come back to cancer biology and you think in the adenoma to carcinoma sequence, which is classical in colorectal carcinoma, we know that the inflammatory Th17 cells are important to trigger malignant transformation, within this adenoma to carcinoma sequence. So we think that in early stages, before the tumor has established, one will come with this TMEM agonist, control Th17 driven inflammation, and there diminish the chances of developing carcinoma. And the other way around is, once the tumor is established, then you have mainly immunosuppressive Th17 cells. So in that scenario, we think that patients would benefit from TMEM blockers and to trigger inflammatory Th17 cells. So from both sides, from anti-inflammatory, and pro-inflammatory, we think that the main thing here, I think, is that TMEM is an emerging target, and we are working on that to trying to show its relevance.
Okay, thanks. And the other question is regarding the effect inflammasome activation. You know that there is some works that suggest that the NLP3 could influence the polarization of T cells directly inside T cells. So do you think, I don't know if you have a look when you check the CD4 T cells, if you check if there is an NLP3 expression in T cells, and if this could influence the polarization of the cells, and correlate these with
Absolutely.
TMEM activity?
Yeah. So the Th17 cells they do express an LRP3, but and they activate inflammasomes. But there is a particularity. This is not through caspase-1. They activate inflammasome through caspase-8. So we went to measure caspase-8 activation both in the knockout cells and by treating them with the inhibitor. And in both cases, we found increased caspase-8 activation when we treated these cells with ATP. So we think that this is part of this inflammatory phenotype too, this increased caspase-8 inflammasome within Th17 cells.
Okay, great, thank you.
Marcelo, hi, Alessandra here. Continues with this question of Vinicius. I'm curious about, do you think that the effect of TMEM, it is affecting cytokines to have this effect in the polarization of the activation or inhibition of the T cells? Because if it is not a transcription effect, what do you think that it is happening? So what the pathways that TMEM is affecting to alters the T cells polarization or activation?
Yeah, that's a very good point, because actually we don't know which are the intracellular mechanisms, by which TMEM is controlling this polarization. So we published last year a work in a transplant model where we can trigger TMEM activity, and this protects, this impairs graft rejection. And in that case, the mechanism involved hemoxygenase 1 expression, this antioxidant enzyme. So we think, and we are working on that now, we think that there will be some redox mechanisms, this altered ionic flux. We hypothesize that this will be controlling redox stress. And well, we are studying now hemoxygenase 1 and redox production, ROS production within the Th17 cells.
Okay, great. This is a new pathway. So, okay, thank you.
Yeah.
Thank you. Very
Thank
good
you. Thank
presentation.
You. Thank you.
So, hi, Marcelo.
Hi.
Very nice presentation, very nice data. I was curious about this TMEM molecule. So, are there any other studies investigating the role played by this molecule in Th17 cells, but in the regulation of immune responses to fungal infections or bacterial infections, or in autoimmune diseases as well? Just to know if in
Yes.
these
So
other
yeah.
contexts, it also has this regulatory effect on Th17 cells as well.
Yes. So in infectious models, we found a role for TMEM in coronavirus disease. So here we worked with a mouse virus, and we also have data on COVID-19 patients. And our main conclusion was that Torid was blocking inflammasome activation within monocytes, and by doing so, it is preventing CD8 T cell exhaustion. So in coronavirus, we think that there is this, anti-inflammatory and protective role of TMEM in that context. There are some other papers linking Torid with viral immunity, and we have shown that that TMEM controls antigen cross-presentation. So by that mechanism, that will be relevant in viral immunity and probably to intracellular bacteria too. I don't know works on bacterial immunity on TMEM, so I think that has not been described so far, nor with fungal immunity. And with autoimmunity, there are some papers linking, a TMEM increase in peripheral cells with responses to immune interventional approaches. So in rheumatoid arthritis, increased TMEM in peripheral monocytes is associated with responses to anti-TNF antibodies. And in multiple sclerosis, type 2 interferons therapy and successful therapy is also associated with increased TMEM in peripheral blood. But there is not so much studies. We are not now submitting a study on human lupus, but well, this is not published yet. And we are working on the autoimmunity models, EAE, and some others to try to show that there is a role for TMEM in autoimmunity.
Right. Thank you very much.
Thank you.
So, any other questions? Okay, so I want to thank you again, Marcelo. Oh, we have one question.
Thank you. Good morning. Nice talk, Marcelo. I just only have two questions. First of all, if we know of the presence of the Th17 in the microenvironment, tumor microenvironment, it's possible to improve the quantity of interleukin-6 and VEGF and new vascularity. After that, metastasis, it was proved or tested this capacity of using this molecule. And another question is, I saw that a major quantity of percentage of CTL4 population. I don't know if was tested also the therapy after the PD-1, maybe the antibody anti-CTLA4 also, if we'll see if maybe it's possible to have a great results. Thank you so much.
Yes, so coming to the first question, absolutely. I mean, the pro-angiogenic mechanisms is one of the pro-tumoral roles of Th17 cells. We didn't study whether we are controlling angiogenesis when we block TMEM, but one would anticipate that this would actually happen. And if this is the case, we may think in combining, anti-VEGF antibodies with TMEM blockers. So I think that that's, from a biological point of view, is absolutely relevant. But well, we didn't study it yet, but for sure the role of these homeostatic Th17 cells and Tregs is to control angiogenesis. So we think that there will be a role there. And moreover, TMEM is also expressed by endothelial cells. So well, we cannot do. We have now the funding and the students and everything, to answer all the questions. But I think that this is a relevant one. And the second one concerning CTLA4, yes, in our previous paper, when we blocked TMEM, we could also improve the antitumor efficacy of anti-CTLA4. And CTLA4 has also been linked with Th17 cells. So, well, but the short answer is yes, TMEM inhibitors do improve anti-CTLA4, and we did that in a melanoma model.
All right, thank you.
Okay, so thanks again, Marcelo, for your talk. And
Thank you. Thank you.